Greetings everyone, I'm glad to meet you again today. As I know you remember, I'm Judith Musevi, taking you through the biology. Our topic was the circulatory system, where remember we had said it has got three components. We said circulatory system comprises of the blood, it has got the heart, and it has got the blood vessels. For today, we shall look at the blood. Now the blood, he said, it is basically a fluid connective tissue whose main business is to transport materials in the body. It is made up of two main parts. That is, it is a solid part and a liquid part. You have got the fluid part of the blood, which is basically the plasma. It makes up 55% of the blood. And then you've got the solid part of the blood, which is the blood cells, which makes up 45% of the blood. Now to look at this liquid part of the blood, that is the plasma. So the plasma is a pale yellow fluid. It contains all the things we expect to find in the blood in their dissolved form. So we find the plasma being composed of water, which is the main component because it is a fluid. It has got these mineral salts in their dissolved form. So they tend to exist in the plasma in the form of the ions, the component ions. It contains the food materials. The food materials will be present in the blood in their simplest form, that is after digestion is complete. So we expect to find the carbohydrates present in the form of glucose. We expect to find the proteins in their simplest form in the form of amino acids. And we expect to find the fats in the form of fatty acids. We also expect the blood to contain the plasma proteins. These are proteins that are found in the blood fluid part, that is the plasma, in very large amounts. The reason why they are called plasma proteins. And there are three main types of them. We have got the albumins, we have got the globulins, and you have got fibrinogen. We shall talk more about them in just a little while. We also expect to find in the blood the respiratory gases. The respiratory gases here specifically, we are talking about oxygen and carbon dioxide. We also expect to find organic waste materials in the blood, where the main waste material we find there is urea, which is a waste material from the metabolic process of the breakdown of the proteins. We also expect to find hormones in the blood and many other things such as vitamins, such as antibodies, and so on. Now for the plasma, basically it has got a pH of about 7.5 in most places. Now, to go back to the plasma proteins that we talked about, that is the albumins, the globulins, and fibrinogen. They are all synthesized in the liver, which is a vital organ, and these plasma proteins tend to vary in terms of size and also quantities that they are found within the plasma. The albumins, to start with, they are the smallest in size. They are present in the blood in the largest amount. Because of the large quantities, their main role in the plasma or in the blood as it is that they help to maintain the osmotic pressure of the blood, and they also act as protein carriers for basically steroid hormones. And then we have the globulins. Globulins are larger in size than the albumins. They are present in the plasma in smaller amounts compared to the albumins. They are also expected to be synthesized in the liver, except a specific type of globulin that we call the gamma globulin. When it comes to the role of the globulins, gamma globulins specifically act as antibodies, and they are produced in the lymphoid tissue. The other globulins, other than the gamma globulins, they act as carriers for hormones, specifically peptide hormones and also mineral salts. Now for the fibrinogen, these are the largest in size compared to the others, and they're present in the blood in very small quantities. For them, while the albumins are helping with maintaining osmotic pressure of the blood and acting as carriers, and they've got the globulins acting as carriers and also carrying around mineral salts and antibodies as well. For fibrinogen, they play a role in the clotting of the blood. For them, they are important in blood clotting, where they tend to form what we call the fibrin threads in the process of clot formation. Now, for the case of the plasma, that's the much you're going to say about them for today. And you now move on to the blood cells. Now, the blood cells, there are three main types of blood cells. That is the red blood cells that are also called erythrocytes. We have got the white blood cells that are also called leukocytes. And we've got the platelets that are also called the thrombocytes. These are the main types of blood cells that we have. Any other blood cell that we have is basically a modification from any of these three that we have. Now, to start with the erythrocytes, or the red blood cells, one thing to note is that all the blood cells are generally manufactured or produced in the red bone marrow. So for the red blood cells, like every other blood cell, they are manufactured in the red bone marrow. The process by which they are manufactured is called erythropoiesis, and this process is controlled by a hormone that we call erythropoietin. For the red blood cells, in terms of structure, they tend to be indented on both sides. A curve on the inside, we refer to that as a concave. So because they are curved on both sides inwards, we now refer to them as biconcave discs. In their young stages, the red blood cells have got a nucleus, but when they mature, they do not have any nucleus at all. Now, for them to have their biconcave shape with even lack of a nucleus, they need vitamin B12, which is necessary for their maturation. Vitamin B12 enables the red blood cells to know what size to grow to and even what shape to take. So it helps them to get the right size and the right shape, which is important in terms of their function. For the red blood cells, they contain a red oxygen-carrying pigment that we call hemoglobin. Now, hemoglobin consists of a protein that we call a globin protein and what we call a heme group. What we call a heme group is a molecule that contains iron in it. It contains iron in the oxidation state of iron 2. We all know iron is an element with many oxidation states, but the one that we find in the heme group is iron 2. Each hemoglobin molecule has got a protein and 4 of the heme groups we are talking about. The heme groups provide the sites where it attaches to when it attaches on hemoglobin. So we say, therefore, the heme groups are the active sites for the attachments. Now, the iron in the heme group, it loosely attaches or forms a link with the oxygen molecule. A weak link, the oxygen can attach on the iron without oxidizing the iron. And the heme group will carry the oxygen on the hemoglobin molecule to the body tissues in this combined form in what we refer to as oxyhemoglobin. The red blood cells will they do the business of carrying oxygen to the body tissues until it reaches a point where they can no longer carry the oxygen to the body tissues. When this happens, we say the red blood cell is worn out. When a red blood cell can no longer carry oxygen, we say it is worn out. So and when it is worn out, it is destroyed in the liver and in the spleen by some cells there we call the macrophages, and the red blood cells are destroyed by the process of phagocytosis. So the red blood cells have got a period of being formed that we call erythropoiesis, and there is a time when they are worn out and they are destroyed by phagocytosis. So they have got a period by which they will exist as they carry out their function and activities. So that period of time from the formation of the red blood cell to its destruction because it is worn out gives us the lifespan of the red blood cell. So for the red blood cells, we say they've got a lifespan of 100 and 120 days, after which they are destroyed in the liver and spleen by phagocytosis. as the red blood cells that are worn out are being destroyed, they have this red pigment that we call hemoglobin. So you expect hemoglobin also to be broken down, and as it is broken down into two waste pigments that we call bilirubin and biliverdin. The two waste pigments are normally put in the bile juice in the gallbladder so that they can be excreted through the body's excretory system. So because they are normally put in the bile in the gallbladder, we refer to the two pigments as the bile pigments. So once the pigment is broken down and the red blood cell is broken down, the waste material is carried away by the blood. But although the red blood cells are formed by erythropoiesis, destroyed when they are worn down or worn out by phagocytosis, we find that the number of red blood cells that an individual has is more or less constant. Now, why this number is constant is because the rate of production of the red blood cells is equal to the rate of destruction. In other words, the rate of erythropoiesis is equal to the rate of phagocytosis of the red blood cells. The role of the red blood cells basically is transportation of the respiratory gases. And we tend to find, because the males and the females have got a different body structure, we have got a difference in the number of red blood cells found in the male and that found in the female. So that generally, the males have got a higher number of red blood cells compared to the female. Reason being that the males have got more of the muscle tissue, which requires energy because it is active. So they need more red blood cells to carry oxygen to this active tissue that is the muscle. The female, on the other hand, have got more of fatty tissue, which is a storage tissue. It doesn't require much oxygen, so they have got fewer red blood cells. Now, having looked at the structure of the red blood cells, describing us by concave discs, this is what the red blood cell looks like. You can see it is indented on the inside. That is what we refer to as the concave curve, and it is on both sides. The reason why we call them by concave discs. All red blood cells are similar, so you don't have different types of red blood cells. Their function, as we said, is basically transportation of the respiratory gases, which is oxygen and carbon dioxide. To look at a brief look of how the red blood cells transport the respiratory gases, we start off with the transportation of oxygen. Oxygen diffuses into the red blood cells across the thin cell membrane, and then it combines with the hemoglobin on the heme groups that we talked about, forming a compound we call oxyhemoglobin. Each of the four heme groups combines with an oxygen molecule in a loose link without oxidizing the ion in the heme group. So we find that the combination between oxygen and the heme groups to form oxyhemoglobin happens in the lungs, where the air comes in with the oxygen for this gaseous exchange to take place. Now we are expecting that the oxygen will be carried to the body tissues by the red blood cell in this combined form as oxyhemoglobin. In the body tissues, we expect the oxyhemoglobin to release its oxygen to go to the body tissues. So now we can represent that in a simple equation where we have a hemoglobin molecule. Because it has got four heme groups, it can carry four oxygen molecules at a go to give us the combined product, oxyhemoglobin, which is the means by which oxygen is carried to the body tissues. And we expect that to happen in the lungs. In the body tissues, we now expect the oxyhemoglobin to dissociate to give us back the free hemoglobin and the oxygen, where the oxygen now goes to the body tissues. So we expect the reverse to take place in the body tissues. The reaction takes place quite easily because the link between oxygen and hemoglobin is a loose link, just like we said. Now, hemoglobin has got a high affinity for oxygen, which is normally shown by the oxygen dissociation curve. And the reason for this is because when one oxygen molecule attaches to one heme group, it causes the protein molecules in the hemoglobin to alter or change their positions, making it easier for the rest of the oxygen molecules to attach. So for that reason, we say hemoglobin has got a high affinity for oxygen. To demonstrate the high affinity for oxygen, we use the oxygen dissociation curve, and this is what the oxygen dissociation curve looks like. The steep part here tells us how quickly hemoglobin can pick up the oxygen. And it also shows what would happen in case you have got less amount of oxygen and you've got more of carbon dioxide, which is the other respiratory gas. Too much carbon dioxide will cause the curve to shift basically to the right, and that means the hemoglobin will take up less oxygen. Less of carbon dioxide, the curve will end up shifting to the left, and it means the hemoglobin will take up more of the oxygen. So that curve quickly demonstrates to us the affinity or high affinity of hemoglobin for oxygen gas. Now, for the transportation of carbon dioxide, hemoglobin is found on the red doesn't have for carbon dioxide. So for the red blood cells to carbon dioxide is assist and brought an enzyme called carbonic anhydrase. So you have carbon dioxide from the body tissues because you are expecting to take place in the body tissues anhydrase. Forms the bicarbonate ions out of the red blood cell and into the carrying the carbon dioxide with it. So for that, find most of the carbon dioxide therefore is transported in the form of bicarbonate ions. As the bicarbonate ions are carried away, the hydrogen ions are left behind, and they can cause the red blood cell to become acidic in nature. When the red blood cell becomes acidic, that can lead to the death of the red blood cell. So we need the hydrogen ions formed from the carbonic acid. Because the hemoglobin goes to the body tissues as oxyhemoglobin, the high acidity in the cell actually causes the oxyhemoglobin to dissociate very fast to free hemoglobin and go and buffer the hydrogen ions. So you end up with the hemoglobin now combining with the hydrogen ions to form very weak hemoglobinic acid. And this prevents the red blood cell from becoming too acidic, therefore preventing the death of the red blood cell. Plasma proteins do the buffering. They end up forming proteinic acid, which is also a weak acid. So we find for carbon dioxide, it is transported in a number of forms. It can be transported in the form of bicarbonate ions, which is the form by which most of the carbon dioxide is transported in the blood. It can also be combined with hemoglobin, although a small amount, and the product they'll form here is carbaminohemoglobin. It can also be transported while combined with the plasma proteins, where the product here they'll form is carbamino protein. And you have a small amount which is transported while simply just dissolved in plasma. And for the transportation of we shall go, and for the red blood cells. Now on to the white blood cells, or the leukocytes. Just like the red blood cells, the white blood cells are also formed in the red bone marrow. They are formed by the process we call a leukopoiesis, also controlled by a hormone that we call leptin. For the white blood cells, we have got different types of white blood cells. And all of them have got a nucleus when they are mature. So they've got a nucleus in maturity. Now for the white blood cells, we have those that have got granules in the cytoplasm, and have got those without granules in the cytoplasm. So we divide the white blood cells into those with granules in the cytoplasm that we now call granulocytes, and those without granules in the cytoplasm that are now called agranulocytes. Agranulocytes meaning absence of granules. For the white blood cells, therefore, unlike the red, we have got different types of white blood cells. Start off with the agranulocytes, the white blood cells that don't have granules in the cytoplasm. For these ones, agranulocytes do not have they have a nucleus that doesn't have any lobes. They don't have granules in the cytoplasm. For the agranulocytes, we have got two types of agranulocytes, the lymphocytes, and to differentiate the two, the lymphocytes tend to be smaller in size. They have got a spherical nucleus that is large in size and almost covers the entire cell. For the monocytes, they have got a bean-shaped nucleus and they have got vacuoles in the cytoplasm. Now for the lymphocytes, something to note, they are formed in the red bone marrow like every other white blood cell, but they can also be formed in the lymphoid tissue, such as the spleen, the lymph nodes, and so on. For the other category of white blood cells, that is the granulocytes. For the granulocytes, blood cells that have a nucleus with lobes in it, so we say they have a lobed nucleus, and they have granules in the cytoplasm. There are three types of granulocytes: the neutrophils, the eosinophils, and the basophils. To differentiate the three, the neutrophils have got 3 or more lobes and then they have very fine granules in the cytoplasm. The eosinophils, on the other hand, they have 2 lobes in the nucleus, so we say the nucleus is spectacle shaped, and they've got larger granules in the cytoplasm. The basophils, they've got a kidney-shaped nucleus and very large granules in the cytoplasm that almost obscure the nucleus. So for the white blood cells generally, looking at both granulocytes and agranulocytes, we end up with 5 different types of white blood cells: the lymphocytes, the monocytes, the neutrophils, the eosinophils, and the basophils. Although the white blood cells are different, they all play the same role of body defense. So to differentiate our white blood cells, we have got, this is what the lymphocyte looks like. No gloplasm, a large spherical nucleus. This is the monocyte, which has got a bean-shaped nucleus. That is the neutrophil with 3 or more lobes in the nucleus. This one has got 3. This is the eosinophil with 2 lobes in the nucleus. And this is the basophil with a bean-shaped nucleus. So just like we said, all of them, they may be different, but they do the same role of body defense. Different, they defend the body in different ways. The lymphocytes will defend the body by producing antibodies. So very important in protecting the body against viruses or other disease-causing agents